Today we will talk about the genetic terminology. We will discuss what is dominant allele, what is recessive allele, what is homozygous condition, what is heterozygous condition, what is gene pool. So, what is dominant allele? Dominant allele is that type of allele which can express itself in the homozygous condition as well as in the heterozygous condition. Or we can say that dominant allele is that allele which can express itself in the presence of the contrasting allele or in the presence of the alternative counterpart. For example, we know that the allele which expresses itself in the heterozygous condition, is known as dominant allele. For example, we know that alleles are always present in pairs. They are present in pairs. If both the alleles of the trait are similar, we say that it is the homozygous condition. That means both alleles of a trait are similar. We say that it is homozygous condition. Or if two alleles of a trait are different or dissimilar, we say that it is a heterozygous condition. We say that it is a heterozygous condition. We know that we have two sets of chromosomes. Chromosomes are present in homologous pairs. These are homologous chromosomes. That means half from the these are one pair of alleles. For example, one chromosome from father, this, and one chromosome from mother. Homologous chromosomes are always present in pairs, and we get one chromosome from mother and another chromosome from the father. See, if we have for example, here on these chromosomes genes are present, and we know that genes are present in pairs. Those are called as alleles. For example, if for example here we will take the example of the T. If allele T, capital T, and here on the another homologous chromosome we have small t, that means this is a heterozygous condition. It's a heterozygous condition, and this is simply genotype. What would be the phenotype? Phenotype would be tall. It means that this is the allele for tallness and this is the allele for dwarfness. T has expressed in the phenotype and it is tallness. So we can say that T is a dominant allele. The allele which expresses itself in the heterozygous condition is known as dominant allele. Or we can say that the allele which expresses its effect in the phenotype of an organism when present in the heterozygous condition, we say that is a dominant allele. This dominant allele, it is shown by capital letter or uppercase letter. So this, what do you mean by dominant allele? The allele which expresses itself in the phenotype of an organism when present in the heterozygous condition is known as dominant allele. Same is the case, we have another. For example, we have this is, for example, this is or allele and this is small or allele. That means here we get round series. Genotype is capital or small r, but capital R has expressed it and we get round seeds in phenotype. So we can say that R has expressed in the phenotype in a heterozygous condition. This is or is a dominant allele. And the allele which is unable to express in the presence of its counterpart, in the presence of its contrasting allele, is known as recessive allele. That means here, if we take this example, T has expressed in the phenotype, while as small t has not expressed in the phenotype. This is a heterozygous condition. And we get what phenotype? We get tall. That means capital T has expressed itself in the phenotype and small t has not expressed in the phenotype. So the allele which expresses itself in the phenotype in the heterozygous condition, it is a heterozygous condition. When two alleles are dissimilar, it is a heterozygous condition. The allele which expresses itself in the heterozygous condition is known as dominant allele, and the allele which is unable to express itself in the phenotype in heterozygous condition, when present in the heterozygous state, we say that it is a recessive allele. So capital T is dominant allele and small t is recessive allele. So what is recessive allele? Recessive allele is that allele which is unable to express itself in the presence of its counterpart, in the presence of its contrasting allele in a heterozygous state. So we can say that it is a recessive allele. So recessive allele is, how we can define it? Allele which is unable to express, the allele which is unable to express itself in phenotype when present in the heterozygous state, when present in heterozygous state. When present in heterozygous state is known as recessive allele. That is recessive allele. So this is this is the case of it is a heterozygous condition. That means two alleles are dissimilar, so we say that it is a dissimilar. Two alleles are dissimilar, we say that it is a heterozygous condition. This capital T has expressed itself in the phenotype, so we call it dominant allele, and this small t, it has not expressed itself in the phenotype, so we call it recessive allele. Same is the case here. We have got this capital R has expressed itself in the phenotype. That is why we get round seeds in the phenotype. So capital R is a dominant allele, and small r has not expressed itself. Small r has not expressed itself in the presence of its counterpart, in the presence of its contrasting allele. So we say that small r is a recessive allele. Small r is a recessive allele. So this was what is dominant allele, what is recessive allele. Then we will discuss, this was dominant allele and recessive allele. Dominant allele is that allele which expresses itself in the heterozygous condition, that is called as dominant allele. And this dominant allele, it is shown by the capital letter or capital Case letter. And in this case we have got, for example, this heterozygous condition, capital T is a dominant allele. Same is the case, capital R is a dominant allele. What is recessive allele? The allele which is unable to express itself in the phenotype of an organism, which does not express itself in the phenotype of an organism, we say that it is a recessive allele. This recessive allele, it is shown by the small letter or small case letter. Remember that the recessive allele can express itself in the pure state or in the homozygous state. For example, we have this, we have small r. That means here the recessive allele, this is a recessive allele, it can express itself in the pure state or in the homozygous state. So we get what phenotype? We get wrinkled seeds. Same is the case, we have small t. That means small t, this is a allele for, it's a recessive allele, it is a allele for dwarfness, but this recessive allele can also express itself when present in the homozygous state or pure state. Homozygous state means, or homozygous condition means, when both the alleles of a gene are similar, when both the alleles of a gene are identical, we say that it is a homozygous condition. So here we have got two alleles are similar, we say that these alleles will express itself in the phenotype, and we get what? We get dwarf pea plants. So this condition is called as homozygous condition. Homozygous condition. So this was what is dominant allele, what is recessive allele. Then what is another is homozygous and heterozygous condition. homozygous and heterozygous condition. Homozygous and heterozygous condition. This is what is homozygous condition? Means when both the alleles of a gene are similar, we call it homozygous condition or pure state. Homozygous condition. For example, we have got our pure state. That means two alleles of a gene or we have homologous chromosomes. These are homologous chromosomes. One chromosome from the father, one chromosome from the mother. This is, for example, capital R. This is capital R. So this is called as homozygous condition or pure state. That means both the alleles are identical. Both alleles of a gene similar. This is what is called as homozygous condition. Capital R. This is homozygous condition. Or we have small r. This is homozygous condition. Or we have capital Y. This is homozygous condition. Small y. This is homozygous. This is homozygous dominant and this is homozygous recessive. This is homozygous dominant condition. Homozygous dominant condition, and this is homozygous recessive condition. This when there are homozygous allele for a trait, they always breed true. That means there would be same trait generation after generation. So we call it as a pure. This homozygous is called as pure because of the fact that they produce the same trait generation after generation. So homozygous condition, it is called as pure. That means they will always produce these individuals will always produce what? Round seeds. On self-pollination, if these individuals are self-pollinated, they will produce what? Round seeds. So we call it the pure state. For example, if I will take, if this is a pea plant, and in this pea plant we have round seeds. We have round seeds. But these round seeds are what? This is condition is homozygous condition. Capital R. We have genotype is capital R, that is homozygous dominant. These two alleles, they will express and they will produce round seeds. And when this is self-pollinated, if this is self-pollinated, flower, this is seed. This is capital R, it is the genotype, and this round seed, it is the phenotype. When it is self-pollinated, we will get individuals. We will get individuals which would have always round seeds. This will get round seeds. So it would be like this, or. That means these individuals, these homozygous individuals, these are homozygous. These individuals are homozygous for a particular trait. When they are self-pollinated, they will produce the same trait generation after generation. That is what is called as they are pure. They breed true. That means they produce the same trait generation after generation. So we have got this individual. It is having homozygous condition for the round seeds. When it is self-pollinated, it will produce progeny. It will produce offsprings. It will produce progeny that would be having all individuals which would be produced from this plant. They would be having what phenotype? Round seeds. So we can say that this homozygous plants, they breed true for a trait, for a particular trait. Then we have got this homozygous condition. Simply means that when there are two similar alleles for a gene, one allele is present on one chromosome of the homologous pair, another allele is present on the another chromosome of the homologous pair, and these both alleles are similar, they are identical. We call it homozygous condition, or we call it pure state. Then we have got another that is called as heterozygous condition. Heterozygous condition. Heterozygous condition means that, for example, we have same homologous chromosomes. Homologous chromosomes always exist in pairs, and one chromosome from father, another chromosome from mother. They are homologous chromosomes. For example, condition would be capital or small r, or condition would be two alleles. This is gene locus, where the gene actually resides. This is capital T, small t. So here condition is capital R, small r, capital T, small t. So if there are two alleles of a gene are dissimilar, they are non-identical, they are different, we say that it is a heterozygous condition. That means heterozygous condition is that where two alleles of a gene are dissimilar. That is called as heterozygous condition. And these are heterozygous for what? These are heterozygous for our seed shape. For seed shape. Or heterozygous for, we can say that heterozygous for height, plant height. So heterozygous condition is two alleles of a gene are dissimilar. Two alleles of a gene dissimilar. This is what is called as heterozygous condition. Homozygous condition and heterozygous condition we have discussed it. Then there is another, we'll discuss gene pool. What is gene pool? Gene pool. how you can define a gene pool? Gene pool simply means that it is the sum total of all the genes, including the alleles in a population, that constitutes the gene pool. For example, there are 100 individuals in a population. Now we'll take their alleles in aggregation, we'll take their genes in aggregation, and all the genes which would be present in the individuals in a population, that constitutes it is gene pool. So we can say that it is sum total of all the genes including alleles. All the genes, sum total of all the genes, or aggregate of all the genes in a population, constitutes gene pool. Sum total of all the genes including alleles in a population is known as gene pool. For example, we'll take human population. This human population, we have hundreds or thousands of individuals in the human population. Suppose this is human population. Suppose this is human population. Individuals would be different because of the individuals in a population would be different because of the variation. Also, they would be similar because of the inheritance or heredity. Suppose these are different individuals in a population, and now we'll see that in these individuals, obviously there would be the genes, and these genes are represented by alleles in a population, or we can say that genes are represented by alleles in an individual. So in an individual we have got alleles because of the fact that we get one allele from the mother, another allele from the father. So genes are represented by alleles in an individual. So there would be, for example, now we'll take sum total of all the alleles, sum total of all the genes which would be present in this human population, in these individuals. So for example, if we represent these alleles by means of the letters, so there would be A in the individual, B, small b, C, D, capital D, capital E, small e. So this is gene pool. This is gene pool. So aggregate, what is gene pool? Aggregate of all the genes in a population. So aggregate of all genes including alleles in, or we can say that aggregate of genes or including alleles in all the individuals of a population. So this is what is called as gene pool. This is sum total or aggregate of genes present in all the individuals in a population. So the alleles which would be present in these individuals, in this individual, in this individual, in this individual, we will take the sum total, aggregate of all the alleles in all the individuals in a population. We call it gene pool. So we have discussed what the gene pool is. Then then we'll talk about the what is Punnett scare. What is Punnett scare? This Punnett scare this is devised by R.C. Punnet, and it is simply a checkerboard which represents the results of the cross. It is a checkerboard, and this Punnett scare. It shows the results of the cross between the individuals. For example, we have this. This is Punnett scare. For example, we have two individuals. This is male individual, male parent. This is female parent. Their gametes. So this is what is called as Punnett scare. This Punnett scare, it shows the allele combination of the individuals, and also it tells us about the genotype as well as phenotype of the individuals. So it shows possible allele combination of the individuals. Combination of individuals. It shows us the possible allele combination of the individuals. For example, this is tall, but it is hybrid tall. Same is the case. This is tall. This is hybrid tall. This is tall, but it is hybrid tall. This is tall. This is hybrid tall. So this Punnett scare, it is used to show the results of the cross between the individuals. Sometimes we make the dihybrid cross, sometimes monohybrid cross or reciprocal cross. It gives us allele combination of the individuals. It makes our calculations easy in the genetics. It is simply a checkerboard which was devised by the R.C. Punnet, and it shows the results of the cross between the individuals. These were the important terms which were used in the genetics. Thanks.